Hi, my name is Jay Nix. I'm a Beamline scientist at the Advanced Light Source at the Lawrence Berkeley Lab in Berkeley, California. We use X-rays to study the structure-function relationship of molecules of biological interest at the atomic level. We're able to use X-ray crystallography to be able to advance sciences that apply to how we understand basic biological principles and help design interactions with pharmaceuticals and other drugs. My background is as a molecular biologist, but while I was in graduate school I learned X-ray crystallography. So what I do now is more for support of the actual biologist, and I do more biochemistry and biophysics to help give them the facilities they need to perform their experiments. As manager of my facility, I basically wear all the hats that need to be worn. I do the biophysics. I will collect and process data for people that need it. I'm also in charge of maintaining and repairing the facility as it breaks. I do the system administration and some of the programming also. But I also have a core group of people that I can rely on to help me when I get in over my head or that the work becomes too much for one person to handle. The synchrotron works by accelerating electrons around a ring, and by the conservation of momentum, whenever these electrons make a turn, it ejects energy in photons as X-rays. They're called light sources because they're actually producing photons in the electromagnetic energy spectrum. We work in the high energy electron range with about a 1 angstrom wavelength. There are so many because the science that you can do with these are so important. And macromolecular crystallography has driven many of these experiments in different places around the world. And it really comes down to these facilities are needed by so many people, and the information is so important that we needed to build out these facilities. And in the late 90s, a big push was made by the Department of Energy to make this available, which keeps America at the forefront of this type of science. The data that we actually get are spots on individual images, and this is because the crystals that are grown interact with X-rays by what's called Bragg's law, and it's a Fourier transform of the electron density that surrounds the atoms of the macromolecule. These spots have information of all the atoms within the macromolecule, and then through mathematics and reverse Fourier transforms we're able to determine the electron cloud that gave rise to those spots, and our understanding of chemistry allows us to place the correct atoms in the correct places and see their interactions in a three-dimensional lattice. And at the resolutions we're looking at within 1 angstrom of stuff, that we are very confident that what we're seeing is where the actual atoms are going, because the electrons themselves, the cloud is much larger than the actual atom position itself. So those differences at the electron level itself are masked by the distances that we're looking at and collecting our data. The synchrotron beam is really nice because it's very, very high brilliance and high flux. So, for example, home X-ray sources can do X-ray crystallography, but they're limited to the amount of time that you can actually have the sample in the beam. So what might take an experimenter from 3 hours to 24 hours to do one experiment, we could do at the synchrotron in 10 to 15 minutes. This increases the output of the science, plus the quality of the data that you get is much higher than what you could get from a home source also. There are complementary methods to growing a three-dimensional crystal, which is often the rate-limiting step of this type of science. What we are working towards now is a complementary method called small angle X-ray scattering, where you can have the molecules of interest in solution and you can look at the molecular envelope. You don't see it at an atomic level, but sometimes the question doesn't require that type of resolution. So you can see two large things interacting with each other, and based on the atomic structure you have, you can model into that and make very precise models of how things are behaving in their aqueous environment. Our samples are sent to us as preformed grown crystals that have been frozen in liquid nitrogen and then sent in cryodewars to keep them cold. We were built by a group of universities, but we also provide at least 25% of our time to general users from anyone in the world that wants to send samples to us to use our facility to understand what's going on with their molecules.